Better now. So I'm going to talk today about a work that we did during the pandemic. So we wanted to see if we are able to repurpose numb drugs for treating COVID. So essentially, drug repurposing means that we are using some drugs that they are numb for treating a disease that is not what was approved to. So a big example is sildenafil, which is known as Viagra, that was first approved to treat CAD, so coronary artery disease. However, when they started the clinical trials, they noticed that actually the males they started having an unexpected erection, and that was considered a side effect. And then the drug was repurposed for treating that disease. Uh, and in the same idea, we can think about many other drugs that can be used for other diseases. That's not what they were developed for. So for COVID, why that was important is basically we started with a disease that no one knew how to treat. What can we do about this? We were everybody locked inside our places, and we wanted to go outside and have our normal lives. However, to develop a new drug for treating an unknown disease, it takes time. It takes a lot of investment, like financial and timely investment. So it's quite expensive. Therefore, using a drug that is already known would be the best approach. And also, waiting for the whole time that for having the vaccine would also take some time. Now we are all vaccinated, so it's not a big deal anymore. Um, so what we decided was to test all network medicine technologies that we had in the lab and known from other labs, and did this big collaboration to identify those drug repurposing opportunities.
Daisy, I'm sorry to interrupt. We're still seeing your first slide in the regular mode rather than the presentation.
Oh,
Can you perhaps double check what part of your screen you're sharing?
yes, definitely. Let me share again.
Okay, so now
Uh,
I see the network medicine title slide, but still in the regular mode, not in the presentation mode.
and now you don't see anything. Huh. Very interesting. Um, Let me stop sharing and try again.
Okay. As
No.
backup plan, if you can't get the presentation mode to work, you can just click through your regular slides.
Okay, let's see. Yeah, so now I'm sharing this screen and
Now we have presentation mode. Perfect.
good.
Thank you. Thank you for fixing this.
No, thank you for letting me know. Um, so network medicine is a whole field inside network analysis where we basically try to use network science to solve medical problems. So, network medicine has already helped us identify, for example, disease module discovery, where you have a bunch of genes that you're connected in the network, and you are able to identify these modules or clusters of genes and how they relate to some diseases, some phenotypes, some pathways. Um, we also worked with drug target identification, where basically you're trying to identify and validate different kinds of drugs or different kinds of targets or different kinds of drug repurposing opportunities. However, oftentimes one drug's not uh, everything that you need for one drug, but you might need a combination of drugs. And network medicine has also helped us identifying drug combinations that can be used to treat one particular disease. And network medicine can also be used for personalized or precision medicine, where you can place one individual in the network context and identify which would be the best drug for for that particular patient. So I've been talking about networks, but I want to make sure we are all on the same page regarding what is a network and how can we identify them. So networks are basically this entity where you have nodes where something is happening and they're going to be connected to something else, some other node, by a link. And those somethings, they can be from multiple natures. So we can imagine computers, for example, that they are connected in the same. network, in the same network, and they're going to have a link between them. However, we can think about this in more social context. So we have, for example, a party where we have five individuals here. Each one are gonna, so a link exists where you talk to another person, whether you're sharing a drink with another person. And then you can see that you have this guy that is the angel guy here. He talks to everybody, he's friends with everybody. And then you have this annoying guy here that he only talks with the good guy. Like he's the only one that wants to chat with him or have a drink with him. In the same way, we can think about our body instead of thinking about like the social context, but like focusing on one individual, how our tissues, how our organs are sending signal to the others. So our brain is sending signal to our lungs, to our heart, to skeleton, to everything, and connecting how they are working together. But if we want to look even deeper inside that, we can look, for example, inside our brain and see how our genes are connected to each other, how they are regulating one another, how they are sending different kinds of signals to one another. Uh, but we can also think about different kinds of network, for example, of virus-host interactions. So here we have an example on EBV. So in EBV, we have in red, those are proteins from the virus that target some human proteins. They are the yellow ones. Those yellow proteins, they also connect to other human proteins. Those are the blue ones, and they can be related to some diseases. So we find here that the activation, or so the proteins that are being targeted by the virus, they can be related to some kind of cancers. And following the same idea, we can think about how can we use this to leverage COVID treatments. So I mentioned that some proteins inside our body, they're connecting to each other. And those are often done using a protein-protein interaction map. So those are constructed either from literature or systematic experimental mapping, basically using hybrid experiment. Um, and we combine multiple data sets here to leverage this whole network. So these networks, they often look like something like this, where you have many interactions happening at the same time. So each one of the dots that we have here, they are one protein. Each one of the links here, they are whether two proteins are binding to each other, so they have the potential to bind. And things that are quite important to remember is that this network is dynamic. So it happens, so many things are happening at the same time, but not everything is happening simultaneously. So this is context specific. So it depends on the disease, it depends on the tissue, it depends on the cell type, and depends on so many factors. But we consider for this purpose up to a certain point a static network, as everything could happen simultaneously. So we know that those proteins that they can be associated to a particular disease, they're going to be in the same neighborhood. So they form what we know as topological communities. And proteins that they are related to the same disease are going to be very close. Proteins that they are related to similar biological process, they're going to be very close. And we can think about this as a city map. So imagine that we are in Manhattan here and we want to go to Broadway. So we want to go to the theater. So we would go to Broadway. We want to go to eat some Asian kind of different food. We could go to Chinatown, for example. But you don't expect to find uh, theater outside of Broadway or something different. So in the same manner, we expect that diseases, genes that they are associated to diseases, they're going to be co-localized. So here we have an example of the asthma disease module. So in pink, we have genes that they are associated with asthma, and in white they are not. So we see that those genes that they are living in a similar neighborhood here. We find also some genes that they are outside, and those could be for multiple different factors, such as we know that the PPI is incomplete. So some of those links, they might not been discovered yet, or some proteins, they can be false positives, right? So we are unsure about they are really associated with the disease. So now we have a big understanding about network medicine and how can we combine all those methodologies into COVID, how this can help us identifying or treating COVID. So we start by collecting the data from the PPI. So we have around 18,000 proteins and 300,000 interactions between them. We also know the targets from SARS, which are around 320 proteins, and they are targeted by COVID when it hits the human body. Oops, sorry. We also have drugs that they come from COVID, from drug bank, and we have 7,000 drugs and around 4,000 targets for those drugs. Here we also leverage three different kinds of drug repurposing prediction tool sets. So one is based on artificial intelligence, another one in diffusion, and the last one on network proximity. We next experimentally validated those results in three different setups. So using clinical trials, using experimental validation in two different setups. So the first one, we send a list to physicians where they chose the drugs that they believe would treat COVID, and another list we screen the drugs throughout, independent of what people could think about them. And later we aggregate those drugs, those rankings in different using different algorithms. So we were talking about disease modules, and one thing that is quite important here is that we are able, when we place targets from COVID in the PPI, we are able to define a very strong disease module, where out of the 300 targets from COVID, 208 they formed this very strong connected component. And I mentioned previously that the modules, they can be disease specific. So what we find here is that there is a module that is stronger in the lungs. So I didn't mention this, but we started doing this work right when COVID started, so 2020 in March. So we were still unsure what could be affected or not. So that was a very strong validation that the methodology really works. So once we defined that there is a disease module, overall, there is a disease module in the lungs, we were very curious to understand which other tissues could also be affected by COVID, right? And we found a very strong involvement of brain, um, digestive system, cardiovascular tissues, and reproductive system. So later we came to know that actually there is a lot of neurological manifestations of COVID, such as the loss of smell, taste, headache, and so on and so forth. We also find that there is a lot of digestive um, system problems such as diarrhea, vomiting, abdominal pain. Cardiovascular tissues also made a lot of sense because we know that patients that they have COVID, they end up having, that they have cardiovascular problems such as any cardiovascular problem, actually. They end up having a higher risk for death. And we also found a lot of reproductive system tissues involvement. And later we found out that actually some of those tissues, they work as bags where the virus used to reproduce much more than others. Uh, so the next step we wanted to know is there any other disease out there that is a comorbidity for COVID, or is there any other disease that is close enough to COVID? So comorbidity is network based, is basically when we measure the separation between the disease module, so how far one disease module is from another disease module in the network. And it two diseases that they have a lot of overlapping proteins or genes, they actually going to be very close to each other. And oftentimes you can think that one drug could be used also to treat this new disease. So our expectation in the beginning was that we would find a disease that was close enough to COVID so we could try to repurpose those drugs. Actually, we did not find anything that was very close, actually overlapping the modules, but we found a lot of diseases that they were close enough. And those diseases, they are also in, they have the same relationship as the tissue. So we found heart diseases, we found neurological manifestations, we found brain diseases, we found lungs diseases here, very similar to what we expect from COVID. Uh, so then, yeah, we have really to go forward to find drug repurposing candidates using network medicine approaches. So we have three pipelines here that I'm going to show. So the first one is based on proximity, where we have three different pipelines. We have network diffusion with 5 pipelines, and we have AI prioritization with 4 pipelines. So the drug repurposing method, basically what you try to identify is once you know where the virus is hitting on the PPI and you know where those targets are hitting in the PPI, which would be the closest path between them. And we obviously measure this also by randomly placing the targets in the network and trying to see whether this distance that you found is there just by random or not. The next one is a diffusion. Oh, sorry, I didn't mention, but we did 3 pipelines based on proximity. And what changes between them is basically the kind of what we are considering a target from the drug. So we can consider any kind of target that is given by a drug bank, or we could also consider only the primary target and or genes that are differentially expressed under the presence of the drug. For the diffusion methods, what we basically do is that we calculate the probability of the target from the drug, from the virus being hit by the target of the drug. And we can recalculate this in different using different similarity matrix. So we use here in Manhattan distance, relative entropy, that is Kullback-Leibler distance, and a JS divergence. Uh, and the last one that we use is the AI-based repurposing, where we create a knowledge graph with basically multiple layers and different kinds of edge types, where we have drugs, we have proteins, viral proteins, and we have the human proteins. And the task that we have here is basically to predict what a disease, a new molecule, what disease the molecule could treat. So we have a ranking for each one of those drugs, for each one of those methodologies. And what we identified is quite obvious at first glance is that the methods, they are quite biased towards itself. So all the AI methodologies, they're going to give you a very similar ranking across them. The diffusion methods, they also very similar across themselves, and the proximity, they are also very similar across themselves. Apart from P3, that basically it's so that proximity tree that is by it's based on differential expression, so it gives a different kind of drug interaction with the targets. So that also gives us the idea that those drugs could be actually complementary to each other instead of competing. So the next, what we did is basically we tried to see how good our predictions were, and we used as the gold standard here clinicaltrials.gov. So we find that AI was doing really well. Proximity was okay, and diffusion was not as good as the other methodologies. However, if we combine the methodologies, we actually have a better prediction than any of the methodologies by itself. So what we learn at this moment is that when we combine all those methodologies, instead of saying like, oh, AI is better, or proximity is better, or diffusion is better, is actually when we have all of them together, we actually have a much better outcome. So what we did next is we experimentally validated those predictions. So we tested 918 drugs, and we found that only 77 they were effective against COVID. We retested some of the drugs. So the top 200 drugs that they showed some positive outcomes, they were retested in different cell lines, and three of those so 4 showed to be effective. So we say 6 here because chloroquine and hydroxychloroquine, they also counted. And 3 of those drugs, they were not in clinical trials. So here we had a very interesting approach to also classify the drugs, because we had to build a classifier for each one of them. I don't think I have time to get into the details. So what we did next was to understand what kind of drugs were we saying that they were actually good or able to treat COVID. What we found is that we have a very broad list that spans from all kinds of drugs, so respiratory drugs, cardiovascular drugs, antibiotics, immunomodulators, proteasomal drugs, and that was not obvious for us at the beginning that would happen, because most of the methodologies for drug repurposing, they're going to be biased towards one class of drug. And actually combining all the 3 methodologies, we don't have a biased methodology, so we can find drugs from all spectrum of different kinds of drugs. So the main points that we found here is that we are able to rank different kinds of drugs to treat COVID. If a drug did not make our list, it doesn't mean that this drug is not effective, but it might be that some of the targets are missing. The PPI is a little bit incomplete. So fixing the data issues could lead to a better outcome towards the end. And another thing is we find a lot of different kinds of drugs here, as I mentioned, but it doesn't mean that if a drug made our list, that drug really can treat COVID, because we know that how the system really works is very different than in a Petri dish. So that's a disclaimer we have to do. So yeah. And lastly, I want to thank the whole team that we worked together for making this project possible. And I'm open to questions, and I apologize for all the technical issues. I cannot hear anything.
Should I try? Can you hear me now?
Yes, I can.
Okay. Did you have a chance to test pairs of drugs, whether they, 2 drugs can attack different parts of the PPI, and maybe together they can be even better than single drugs?
So we started a project working on drug combination, but we did not move forward because to validate that, we would need basically to be able to test all the drugs against all the drugs. And just selecting a couple of them would be very hard to validate and say like, yes, our methodology is really doing pretty well, but we don't know the negative outcomes here.
Thank you very much. Any other questions?
Really nice talk, Deisy. Thank you. I wonder if you do this analysis on a random graph as a negative control. Did you try that? Like, would you get some of those drugs even the PPI is just a random PPI?
So we don't test against a random PPI, but we test always against random targets.